Hello and welcome back to Core Science. In today's lesson, we will be looking at the science behind this particular headline, which is the coronavirus tests. So the question that I want to answer is, how does coronavirus testing work? And I could give you a short answer for this question right now. And essentially, when you do a coronavirus test, you are looking for the genetic material of the virus. So if you remember from last lesson, we looked at the structure of a virus briefly, and it essentially contains the genetic material, so DNA or RNA, and then it's encompassed in a protein code. So when you test for the virus, essentially you're testing for the genetic material of the virus. So in order to understand, I actually can't spell. In order to understand how you could potentially test for the genetic material of a virus, and you could test for the genetic material of a lot of organisms, like human and, well, every organism really. So in order to understand how you could do that, we need to understand the structure of the genetic material, and the genetic material that we will look at specifically in this lesson is going to be DNA, even though it's in the case of coronavirus, it's actually not DNA that's being tested, but we'll talk about the other types of genetic materials later. So the agenda for this lesson is to be able to identify the basic components of DNA, and then I'm going to explain what a nucleotide is, and then we'll describe the purpose of complementary base pairing in DNA, and then use complementary base pairing to predict the sequence of DNA strands. So let's just go back to the analogy that I made last lesson, which is this is a representation of the cell. And if this is a representation of the cell, then this is where you would find DNA, which is inside a nucleus. And DNA is essentially just a set of instructions on how to make proteins. And if you do a quick Google image search of DNA, which is what I did. These are some of the photos that I found. So I'm sure you have seen this before. When we talk about DNA, this is a very common representation. So it looks like a twisted spiral structure. This one's a little bit fancier. You can see some of the components. So there's some bits in blue and then red. And then there are actually a bit here that's green and then yellow. So you can see the different components being color coded in this photo or picture. Now this one is another representation. This is not what a DNA is supposed to look like, but the 1 and 0 is essentially binary coding in computing. So it's trying to indicate the function of DNA, which is to code for something, and that something is proteins. This one is even better because it shows you what the code in DNA actually looks like. So it's not a series of numbers like in computing. It's actually a series of letters like A, T, C, G, and we will look at what they mean in a bit. And then I found this image, and I'm not even sure what it's trying to say. I think this is too artistic, too out there for science, because the DNA strands kind of disappear. But anyway, so you can see a variety of images trying to represent DNA, or just take that unique helical double helix structure of DNA, and then they can do some really interesting things with it. But if you need to have an actual definition, then DNA actually is an acronym. So it is an abbreviation for deoxyribose nucleic acid, so D, N, and then A. And it is a type of biomolecules found in all cells. So once again, if you recall from our last lesson, we looked at 4 types of macromolecules or biomolecules, and DNA is one of them. And essentially what it is a set of instructions on how to make proteins. So I used to have this teacher, and he used to say that DNA is not that interesting at all. It doesn't really do anything. It it's just there as the instructions. However, without the instructions, you can't really do anything. So I guess it's still very important. It's just it's not very involved in the action. It is often compared to a set of blueprints, and it is unique for an organism. So my DNA will be different and unique and from your DNA. So that's good. Unless you're an identical twins. In that case, someone else will have the same DNA as you. Okay. So there are so many photos of these on the internet, and this is the art and craft section of today's lesson. Somebody made an origami from one of the instructions that I showed last lesson, which is really good. So if you really want to, you could build this if you have enough sweets at home, and you can convince your family to allow you to use all of those sweets to make a model. If we were in class right now, we would have built this model, and then you would get to eat the sweets. But unfortunately, we're not in class. So this is a very good representation of DNA though, and you can see some others, like you have licorice and marshmallows and lollies. Anyway, this is actually better because it actually shows you the component even in this part rather than having a continuous chain. So if you look at this particular model, then the first thing you can see is that DNA contains two strands. So this is one, and then there's another, and they wind around each other like a twisted ladder. So a ladder that is twisted is the analogy that we use a lot when we describe DNA, and you can see the steps here. And this is called a double helix. So a double helix is just referring to a twisted structure. Now, when you have to draw it, this is actually what we tend to draw, which is not that accurate. I guess this is a lot better, but it's really hard to fully represent this three-dimensional structure in 2D. So I would strongly encourage you to draw this to try to build this today if you have the tools. Now, if you have some plasticine like Play-Doh, you can, and some toothpicks, that's enough as well. So if you untwist that ladder and then pull the two strands apart, you will see that there are two strands here. I have to enlarge this image. I do want to make a disclaimer. I did not make this model. I got this off the internet. I have no sweets in my house to make anything. But you will see that each DNA strand, so there are two strands in a DNA molecule, and each strand consists of a sequence of molecules called nucleotide. So you can see some repetition here. So this is the first nucleotide, and then this is the next one, and then this is the next one. So nucleotide. Okay, so you have multiple nucleotides strung together, and that gives you one strand. The nucleotides also have a general name, and they are called the monomers. Mono is one, so monomers is like one of the parts, and multiple monomers string together will give you what is called a polymer. So a DNA is a polymer, and poly just means many. So a polymer means something that has multiple parts, and each part is called a monomer. So you have 5 monomers on this side, and then you also have 5 on the other side. I'm not going to go into this at all, but the other strand is actually going to be in the opposite direction. So this one is this one is upside down. I'm not going to explain why. You don't need to know for core science, but I think if you do biology or forensic, I'd say, you would be able to explain why. Okay, let me just finish annotating all of the monomers. So you have the same number of monomers on each strand. So these are the nucleotides. So each of the parts, so if we pull all the parts apart, you will see this. This is a nucleotide, and every nucleotide, if you see here, and then you can go back to the previous slide, this thing is cut short, but it does have a bit here. Every nucleotide contains 3 parts, if you see, and it has a very, very specific shape. So the 3 parts are, you have a sugar, so this is the sugar, and the sugar is going to bind to another bit on top called a phosphate, and then on the other side the sugar is going to bind to what is called a base, or a nitrogen containing base, or an N base. So this is the base. So there is no specific connection or direct connection between the phosphate and the base. They all combine through the sugar. So this is another representation. The sugar, and obviously when you make a model, it's really hard to represent the shape correctly, but the sugar in DNA is actually a pentagon. So this is your sugar. It is called deoxyribose sugar, but you don't need to know what deoxyribose mean for year 10. And then this is the phosphate, so we tend to put a P here, and then this is the nitrogen containing base. But we normally don't put an N here, and the reason will come clear in a second. So that's another, this is another representation. So sugar, phosphate, and the base. So three structures, a very, very unique shape. And hopefully another thing that you can see, and I hope I have it here. Oh, no. Hopefully you can see in this photo that the sugar and the phosphate, so every nucleotide has the same sugar and phosphate parts, but there are 4 different colors for the bases. So you have green, or orange, or yellow, or pink. So 4 different colors. And this is to represent the 4 different types of bases. So they all have the same sugar and phosphate groups, but they can vary in the base that they have. And there are 4 types of bases. You do need to know these names. Adenine, normally abbreviated to A. Thymine is T. Guanine is G. And cytosine is C. So if I draw an adenine nucleotide, the sugar will be the same. And then you have, okay, let me draw a better pentagon. So this is the sugar, and then there's the P, and you probably would put an A here to show that it is adenine. So you don't put an N for nitrogen base, you put the actual base out of these 4 letters that the nucleotide has. There is a Center for Genetic Research in Melbourne, I think near Carlton. It's actually called the GTAC Center. I don't know whether any of you have been there, but the name, it's taking inspiration from the 4 bases of DNA. And then there's a movie called Gattaca. I hope this is the actual spelling. And that movie also talks about a world when we can genetically engineer our gene and DNA. And it, once again, the title of the movie came from the 4 bases. Now, in this particular case, and I have specific reasons for this. This is not just a coincidence, but I'm gonna say that the adenine is the orange one. So this is A. The thymine is yellow. So this is T. The guanine is green, that's easy to remember, and the cytosine is pink. So you have 4 different types of bases in this model. This is what it actually looks like. It looks a little bit terrifying. This is just for your information. You don't have to remember any of these, and I don't think you have to do that at all until you get to university if you want to study biology or biochemistry science. Okay, so let's just go back. The DNA strands, we've talked about this before, but to form a strand, the phosphate, and I kind of reorient the photos, but you can probably see that this is the sugar and this is the phosphate and this is the base. So the sugar, the phosphate of one nucleotide, so this is one nucleotide. The phosphate of one nucleotide, I think that's one nucleotide. No, sorry. This is one nucleotide. So it has the correct orientation. So the sugar of one nucleotide is going to bond with the phosphate of another nucleotide, and that creates the backbone, if you will. So this bit is the backbone. So this creates the backbone of the ladder. This is also called the DNA backbone. Now the bond between the sugar of one nucleotide and the phosphate of another nucleotide is really strong. So this is a much stronger bond than the other bond that we'll talk about in a second. So you have a backbone from the sugar of one nucleotide and the phosphate of another. So this is one nucleotide. And then let's say we have another nucleotide here. So a DNA strand is constructed just by having the sugar phosphate bond here. And then a strand is unique due to the type, number, and sequence of the base. So if we look at here, so adenine is orange, so that's adenine. Thymine is yellow, probably will not write the word base here. So thymine, and then you have guanine and cytosine. So this strand, even though both strands have the same number of nucleotides, these two strands are different from each other because one of them will have the sequence of T, C, G, A, C. That's just the nucleotide base sequence. And then the other one is G, T, C, G, A. So that's the other one. So they have different types and sequence of nucleotide bases, and that is what is unique about them. Now, when you have the two strands, remember that a DNA contains two strands, and the two strands have to be joined together, and this is the function of the base. The bonding between the base of one strand and the base of another strand is what joins the two DNA strands together. But if you look at this, this is essentially the two strands from before. Hopefully you can see that they have very, very specific pairing. So all the green or guanine will always bind with the pink or cytosine bases. And then I can't remember what the other one was. Orange. Yeah, orange is adenine. Adenine will always bond with yellow or thymine. So this is what is called complementary pairing or complementary pair. And the bonds between the two bases are weaker than the bonds in the backbone. And the way the complementary base pair works, as I just mentioned, is A always joins with T and C always joins with G. So if we look here, we have a GC and then TA and then CG and then GC and then AT. So you will never have a situation when the A joins with the C, or the A joins with the G, or C joins with T, or C joins with, oh, actually, I think that's it. And G cannot join with T either. So you will never have these pairing. It's always A to T and C to G. That's a very, very unique part of DNA, and this is complementary pairs or complementary pairings. So there's an acronym, or not an acronym, a mnemonic that you can use to remember this pairing. So adenine forms a bond with thymine, or apple and tree. So A and T go together on apple tree. Just know apple tree is probably enough. And so adenine to thymine. And cytosine bonds with guanine, so car and garage. So apple tree and car in garage. I'm sure there are other mnemonics that you can use to remember the ATCG relationship. This is another one that someone told me recently, so you can use it if you want. So GC is George Clooney, G to C, and AT is Mao. Oh no, I think I'm gonna pronounce. I think this is not. Is it? I think it doesn't have an L here. I'm sorry, I don't remember her name. And the twins. So AT. I thought it was pretty good if you like George Clooney. Anyway, so apple tree, car in garage. And this essentially forms the foundation of what is called DNA sequence. And I actually already gave you an example of DNA sequence before. It essentially is a sequence of the bases. So things like A, T, C, C, G, G, T, something like that. That would be an example of a DNA sequence. And your DNA sequence would be different from my DNA sequence, even though our DNA would be made of the same nucleotides. It's just how many bases we have and what is the order. And 4 bases are enough to form a variety of different sequences. The interesting thing about DNA, though, is due to the complementary nature of DNA, you just need to know the sequence of one strand to work out the sequence of the other strand. So there are 2 strands in DNA. Let's say one strand is has the sequence of A, T, C, C, G, G, T, which is what I just wrote here. You can work out the sequence of the other strand just by this, because you know that they can only bond together via complementary pairing. So let's just use the other strand. So A can only go with T, apple tree. So if this is A, that's a T. If that's a T, this is an A. So apple tree. And that's another T here, so that's another A. And car in garage. So if this is a C, there's a G, and then there's another G, and then there's another C, and then another C. So if I know the sequence of one strand, I can work out the sequence of another strand. Now, I just want to, this is again for your information, but the Human Genome Project, which started and ended before you were born, it started officially in, I say, 1995 or 1999. I think that was the actual starting point. It ended in 2003. Maybe it was later. I actually can't remember. I don't think this was the actual start date, but it supposed to run on for 10 years at least. And I don't think this was the actual end date either. But anyway, it was an international project, and ended at the turn of the new millennium. And it was to sequence the DNA of humans. That's why it's called the Human Genome Project. And it took people a decade, if not longer, and international efforts of scientists around the world to sequence the 3 billion bases of human DNA. So that took about 10 years. But then after that, we have things like 23andMe, and I don't know whether any of you have heard of it, but it is essentially a kit, and then they will send you a kit, and you can take, I think it's a saliva sample, and then you send it back, and they would sequence your DNA for you, and it allows you to find out a lot of information about yourself really quickly. So we have gone really far from taking 15 years to sequence a human genome to having kits like this. Oh yeah, it is a saliva collection kit where you can get your DNA sequence, and the problem of course is, even if we have the DNA sequence, it's very hard to know what it means. So you need somebody to run database to translate that sequence into specific information, which we will talk about in a future lesson. But I think we've come very far, and I'm pretty sure there's still a challenge when people are trying to design a technique that would allow you to sequence human DNA in a 24 hours, I think was the time limit. And that would be great, because if it could be sequenced really quickly and really accurately, you can get a lot of information, and maybe you know whether you're at high risk for a condition or another condition. And then we'll talk about the ethical issues that come with that as well in a future lesson. So we will go back to coronavirus testing, because there is a genetic material in the corona. Essentially what they do is they're going to sequence this. So let's just say the sequence is ATTCG, and this is not the actual sequence. It's not going to be 5 bases long. It's supposed to be a lot longer than that. But what they do is, because you just need one sequence, and then you can design this molecule. So I'm going to design this molecule. This is what is called a primer. By the way, you don't need to remember coronavirus testing science for the cat or anything. I just thought if you're interested. And you're going to attach, if you will, like a flashlight or a signal on this primer. So if there is complementary pairing on this primer, it is going to flare up. So because there's pairing here, you can see the signal, and normally it's in the form of fluorescent light. And then what you do is, let's say you have somebody who exhibit coronavirus symptoms. What you're going to do is you are going to take their DNA. So you take their DNA. That's why you take a saliva swab. And then their DNA has like a lot of, it's really, really long. But what you're going to do is you're going to put this primer into their DNA, or into their cell, really. Let's say this person actually is infected with coronavirus. So they have their DNA, and then there's the coronavirus DNA in there, and they both will be in their sample. So if you put the primer in to the sample, so you put this in here, this thing is going to bind to this piece of coronavirus DNA as well, because of complementary base pairing. So every time that this primer sees this sequence, it's going to bind to it. And every time it binds to it, it's going to give out a signal. And it can only bind to the sequence that it is complementary to. So you can see the signal and you're like, yep, this person is positive for the virus. I am skipping so many steps in the actual testing, but this is the gist of it. That's it. That's it for this lesson. I have a quick glossary here if you want to update your glossary list from the last lesson. Thank you.